We can move to the second lecture. Then we move to another group of bacteria, gram-negative bacteria that can cause disease when we weak. But normally in healthy person, they not cause disease. They kind of spy. They are thief when they found that your immune collapse, immune disorder, or you get some immunosuppression, then they will cause disease in your body. Okay. The key bacteria is family Pseudomonadaceae. That means Pseudomonas is the key. And the other bacteria, actually there are some, but I just pick up few of them to show you. Let's start from the idea of what is the opportunistic infection. Opportunistic infection refer to microbe that normally does not cause disease in healthy. Okay, they cause disease in the body's immune system is impaired. Who get this? The first one, people who be acquire immunodeficiency syndrome, even by the other action or get infected with immunodeficiency virus, including HIV, and there are the other bacteria. There are the other virus also. Neutropenia. Neutropenia is it's time when the neutrophil drop in your blood circulation, so the defense is weaker than normal. Or probably people who get organ transplantation, they probably need to provide the immunosuppressor just in case that you will reject graft or graft rejection. Okay. And people whose defect in immune response, for example, diabetes mellitus, to get a target of low immune, or cancerous, the CA, okay, for cancer patient. For particular patient who get infected with HIV, we learn a lot from this group of patients. We never know there are so many OI. We thought that lots of OI have been gone from us in human population for a while. But after we got HIV, we know at least tuberculosis, we know some fungi, for example, yeast, some bacteria, for example, some viruses, for example, cytomegalovirus, also a causative agent of OI, opportunistic infection. But in this lecture, we focus on bacterial opportunistic pathogen. Okay, so do not underestimate of OI, because OI is very important and can make your life threatening, particularly HIV patient. If the CD4 level is dropped, that's make OI can overcome your immune system. So the prophylaxis is maintain the CD4 level. And treatment option, if OI develop, not HIV, sorry. After recover, you need to maintain the treatment for a while until the CD4 back to the general level. General level, not general of healthy, general of HIV patient level. That's still. So let's start from the first family, Pseudomonas, Pseudomonadaceae. The Pseudomonas general are aerobic, okay? Non-spore forming, they can multi, they can move by polar flagella. So Pseudomonas normally are natural habitat of Pseudomonas, water, soil, or everywhere that contain a lot of moist, okay? Pseudomonas itself, they can be pathogen to plants, can be pathogen to human, can be pathogen to animal. And some species move to the new genus, for example, Burkholderia. Burkholderia pseudomallii. Have you heard about this bacteria? Long time ago, this bacteria was Pseudomonas pseudomallii. So then we proved that it is not Pseudomonas anymore. Then we moved to Burkholderia. Mm-hmm. The key bacteria that is very significant in terms of medicine is Pseudomonas aeruginosa. They produce a blue pigment, okay, and get opportunistic pathogen, okay, okay. Pseudomonas aeruginosa is slender gram-negative multi. They not produce capsule, but they produce slime layer. So slime can leak out. So once they produce slime layer, that means they have chance to generate biofilm. Okay, and then biofilm is a weapon of Pseudomonas. Generally, Pseudomonas aeruginosa don't have the very harmful machinery or virulent factor. They have weak or mild virulent factor. So that's why they cannot cause disease in healthy, okay? But the serious thing is, once they get infected, they resist almost all antibiotics. That's the key, makes Pseudomonas aeruginosa serious, okay? They produce grape-like odor, smell like grape, okay? But if you have chance, do not open the plate and smell it. It's not a good idea. They grow aerobically. Some strain of Pseudomonas, or most strains Pseudomonas, can produce beta hemolysis on blood agar like this, but the zone is small. Okay, they produce so many pigment. Okay, can be red pigment, can be green pigment, blue pigment, and also pyomelanin, black or brown pigment. Okay. So biochem of Pseudomonas is alkali slant, alkali butt. Okay. Pseudomonas use glucose by ED pathway. Still remember, not EMP pathway. They ferment glucose by oxidative. Okay. So these are general biochem test: catalase positive, oxidase positive, arginine test positive. Okay. Pseudomonas itself resistant quite a lot. They can grow to so many chemicals that used for clean surface. If you want to kill them, use 55 degrees Celsius for 1 hour. Dettol as a selective medium. Okay? Pseudomonas can grow in Dettol, but the other bacteria cannot. Okay? That's a serious thing. Pathogenesis of Pseudomonas aeruginosa, they infect wound. Once they infect wound, they cause gangrene. When you hear the word gangrene, you probably think about Clostridium botulinum, right? So the gangrene produce colors, the green or blue colors in the gangrene. Okay. That's characteristic of Pseudomonas aeruginosa. Normally, they will not cause serious problem. They can infect eyes, infect wounds, infect burns. Burn is very important. If you get burned and then go to hospital, you need to stay in hospital for quite a while. Then burn can be harmed by infected of Pseudomonas aeruginosa. Another one is the respiratory tract, particularly associated with patients who get cystic fibrosis. Well, the antigenic structure, they process pili or non-pili adhesive. They have so many proteins they attach. They produce biofilm that's even make cystic fibrosis patient worse. LPS, endotoxin, they have so many protease enzyme, okay? Serine protease, metalloprotease, alkaline protease, they help each other damage tissue in the area they infected and help bacteria spread itself to the other tissue. They have phospholipid C that lyse the blood cells, exotoxin A, exoenzyme S and T also that's damage hosts, okay? Pseudomonads very serious for people who get infected, who need to stay for a long time in hospital, particularly in ICU. And patients who use the respirator or endotracheal tube, who use tube, they contaminate with pseudomonads very easy. Okay. Apart from OI, pseudomonads will be nosocomial infection. Normally, you not get infected at home. You probably get infected when you stay long time in hospital. Okay. Apart from pseudomonads, we probably have Burkholderia, okay, Acinetobacter and Moraxella. Let's start from Burkholderia. Burkholderia is saprophyte itself. Saprophytes mean like they grow on a dead plant, dead animal in soil, in pond, in rice paddle, in the area that grow vegetable or plant, particularly in Southeast Asia. It's very important, particularly in Thailand and North Australia. I think more than 90% of cases in the world come from Thailand. This is important. We know Burkholderia that is important for human pathogen is Pseudomallei. Here, Burkholderia mallei mostly infect animal that also harm animal, particularly livestock. Burkholderia cepacia also. The species that is not infected human or harmless to human is Burkholderia thailandensis. Okay. They cause disease called melioidosis. Actually, it is acute pneumonia. And then when you, they can latent in your body. When the disease gone, recover, and then disease come back again, we call relapse. Okay. So, Burkholderia pseudomallei. General characteristic in the gram, it is bipolar stain. This is bacilli, but they stain just only polar. So you probably look like safety pin, like this bipolar stain. Okay? Cells quite small, obligate, aerobic. The colony, they can grow on selective medium called Ashdown agar. Ashdown agar's color is like this, but this is not blood agar. In Ashdown agar, the colony look like cornflower head. Here is wrinkle. Okay? It smells musty, smell like soil after raining. Okay. Oh, another bacteria that process the colony like this, you probably get confused, is Pseudomonas susuri. Can process the colony like this, but you probably need to be careful. Burkholderia pseudomallei process this morphology on Ashdown media. Biochemical reaction for Pseudomonas, for Burkholderia pseudomallei is catalase positive, oxidase positive, and they resist to colistin, and then multi-biflagella. They spread by polluted environment. that contains so many bacteria, and they associated in soil. If patient came to see doctor, probably it is so hard to find the causative, like Burkholderia. The bacteria grow in media so slow. You probably spend like 2 or 3 days to see the colony. But asking patient directly is help. This is one of the febrile illness. Febrile illness mean the disease that get fever but with unknown cause. So if patient occupation is farmer, they kind of plus for melanosis. In Thai, we call Mong Klo Thiam. I don't know what does it mean. That's the name in Thai is so confused. This bacteria can be intracellular bacteria or live extracellular, live intracellular, both. Okay, they can invade into body and go to survive intercellularly. Okay, so you get Burkholderia by skin abrasion or ingestion. You eat contaminated Burkholderia or inhale. This is very important. In the old day, people thought that they quite a lot in the northeastern Thailand, but actually throughout the country. So disease can infect people who some kind of sport like golf player, because when you play golf, you probably hit the golf and you spread some soil, the surface of the soil, and then you probably by chance inhale of the small particle, dusty particle, then you get infected if that area contaminated with Burkholderia. Okay. So then we move to genus Acinetobacter. Acinetobacter is Coccobacilli. Okay. Sometime you probably see its COX signs, but it is classified in the group of gram-negative bacilli. Okay? The key or problematic species that cause serious problem in healthcare is Acinetobacter baumannii. They contaminate with the surface, the soil, sewage, water, and hospital environment. In hospital, mostly when you swab the surface, you probably get Acinetobacter baumannii. So same, I make 3 golden star. This is multi-drug resistant. So they resist to so many antibiotics and cause hospital-acquired infection. Okay. So basically, we use biochem tests to differentiate them. Oxidase negative, they probably show hemolysis, indole negative, catalyst positive. And typing of the A bomb normally use molecular typing, DNA typing by AFLP. AFLP is you just digest DNA of bacteria with restriction enzyme, okay, and link by adapter. So then when you digest DNA of bacteria, you get tons of pieces of enzyme, of pieces of DNA. You link those DNA by adapter A and adapter B. Adapter A and B are different. A piece of DNA linked to the DNA of A bomb, and then PCR by using adapter A and B as a primer binding site. If some pieces of DNA that's too long for PCR, then you probably not observe that, but just only the appropriate size of DNA can PCR. Then you run electrophoresis to separate AFLP band, then we call the fingerprint, or you probably amplify some region and cut that PCR product by specific enzyme we call PCR-RFLP, or DNA sequencing also help for identifying a bomb. Nowadays we shift to we potentially use the whole genome sequencing to study that, because once you sequence the whole genome, you know DNA sequence that can tell this is a Baumannii indeed, or you can tell this is which group of a Baumannii, and also you know which drug resistant, antibiotic resistant gene that's maintained in the chromosome. So you probably know the typing of a bomb, the drug resistant, that help a lot for doctor to design the antibiotic. Okay. So a bomb, now we have MDR, Acinetobacter strain, spread in hospital since 1970. So this is like 50, half of century that we know A bomb is very serious, and the serious case increase every year. Okay, so we need to concern of the A bomb. They dangerous for people who's immunocompromised patient, particularly DM or patient in ICU, cancer, something like that. They can lead to death if the patient is not receive aggressive treatment, because they kind of patient need overuse antibiotic to protect that. So A bomb survive with minimum need. That's mean like they no need media, whatever. They stay on surface for months. After 3-4 months of contamination, you still observe. You can isolate A bomb from the bed rail here, because the patient touched bed rail a lot, and then they probably get infected. Bed clothes and doorknobs or sink in the bathroom, but not in your house bathroom, but in hospital bathroom. That's quite important. The reason they resist antibiotic because they have so many aminoglycoside modifying enzymes. So this enzyme change aminoglycoside antibiotic to less action of the antibiotic. They have broad spectrum of beta-lactamase, so all penicillin group is not work. They have carbapinamase, pidamase. So carbapinam cannot use well with A bomb. Actually, this is drug of choice. Carbapinam is work well in the patient who get infected with A bomb, but still, even we know that they still have the number of resistant case increase, but we still use carbapinam. Okay, they change outer membrane porin to pump the drug. They have penicillin binding protein change. So this is important statistics. Actually, it's not so new. It's like 5 years. In 2015, the incident of carbapinam resistance. Okay? If you see, Thailand is one of the very serious target because it's very deep red color. China, Thailand, India. So we overuse carbapinam, Vietnam, Laos, Southeast Asia, they are so serious. In the US also, in Latin America. Okay. The second serious one is Russia, then Canada, some country in Europe, Japan is, you know, like the incident is lower. Okay. Then we move to the genus moraxilla. Moraxilla is, if you not use biochem test and observe just only gram stain, moraxilla is look pretty much like gram negative cocci, but actually diplococci, and you probably confuse with Neisseria species. It is obligate aerobic, okay, non-motile, oxidase positive, and cause disease in so many in respiratory system, in middle ear, in eyes, central nervous system, and joint. Okay. So bacteria are non-hemolytic on blood agar. They need supplement a little bit by carbon dioxide. Colony is not small colony, but not super small like Helicobacter, not like Haemophilus. Okay, white colony. They cause media otitis. This is main cause from this bacteria. They can cause pneumonia, bronchitis, or even bacteremia. But keep in mind, this bacteria is opportunistic infection. That's why they cause serious problem with, you know, only case that weak in immune response. So in this group of lecture, we will ending just quite few. You probably learn in textbook. In textbook, they talk about a bomb for one paragraph, and there are so many opportunistic infection. Well, opportunistic infection, they're not caused by gram-negative bacilli only, but they have some yeast, some virus, something like that, or some protozoa. All right, that's end of the lecture. Today is so quicker than the other. So you learn all gram-negative bacilli already. Okay. Tomorrow we will spend some time on spirochete. Spirochete, I point just 2 or 3 genus and very little touch on anaerobic bacteria. Actually, anaerobic bacteria, some part of them you learn in spore formation. That's Clostridium, right? So this anaerobic bacteria, just 1 or 2 species about non-spore forming anaerobic bacteria, okay? And unusual bacteria, the reason that I say unusual bacteria because some of them no peptidoglycan. So once then they have no peptidoglycan, they live in their own fashion. They cause disease, and penicillin that inhibit cell wall is not work with them. Okay, that's one group. We refer to mycoplasma, ureaplasma. And another 2 group is bacteria that behave itself like virus because they are obligate intracellular parasite. They cannot live outside the cell. You cannot streak plate. You cannot perform the general bacteriology laboratory technique in these 2 group of bacteria. You probably culture bacteria based on the cell culture of animal and infect like viral infection. They penetrate inside the cell and then they grow. That is rickettsia and chlamydia. Okay, but rickettsia can divide to be so many groups, and some of the rickettsia now move out of rickettsia and form a new genus like Orientia. That's the thing you learn tomorrow. Okay, that's all for today.